One of the things I thought you could help us understand is a little bit of the history of antipsychotics. There have been several generations. I'm not sure if we're calling Brilaroxazine the fourth generation or
No, we
third
call it
plus.
as the next generation.
Next generation, okay.
Yeah.
But there are three generations of antipsychotics.
Yeah.
Maybe you can run us through, just real quick, not too long, kind of the differences and kind of how we started and what each generation represented as an improvement over the previous
We don't call even as an atypical antipsychotic. We call it as the next generation.
Next generation, sounds good. So what was the first generation all about? How
They are D2 antagonists like Haldol, haloperidol. It's even now widely used.
Okay, and we've totally.
The differentiation is they are primarily dopamine active, lack of serotonin. Patients were stabilized by addressing the positive symptom, but difficulty in recovering other symptom domains.
Okay.
The newer drug got approved over the last 40 years, the 13 new drugs.
And that's the second generation?
Second generation.
Okay.
Yeah. Atypical antipsychotics.
Okay.
The second generation antipsychotics, besides the dopamine activity, they do have serotonergic activity, primarily serotonin receptor 5-HT2A. But other receptor activities not necessarily equally potent. That's why they are better than first generation antipsychotics. They are like haloperidol is a
What about the side effects, second generation? Did that improve too, or did that?
So they have a slightly different. If you look at the first generation antipsychotics like haloperidol, dopamine antagonist, a lot of extrapyramidal symptoms.
Okay.
Weight gain and other things are not that high as compared to second generation.
So second generation actually had more side effects.
Second generation antipsychotics have slightly reduced extrapyramidal symptoms or a CNS side effect. However, second generation antipsychotics have higher metabolic side effects.
I see.
So that is derived to cardiometabolic and metabolic.
Okay.
Like these are derived from adrenergic activity and then some of the serotonergic off targets such as 5-HT2C and then muscarinic receptor activity as well. That lead the activities for these off targets lead to side effects. And in general, we club together metabolic side effects. So overall, there is a trade-off. Let's see, in the first generation, are potent for the positive symptom. Second generation, by and large, addressed positive symptom. To some extent, addressed negative symptom mode, but still there is significant unmet need. But they do have a lot of side effects. So the next generation antipsychotic should address these unmet need. It should address efficacy standpoint, major symptom domains, such as positive symptom, negative symptom, mood, and then cognitive. These are the four major symptom domains of schizophrenia. The last one is neuroinflammation. Neuroinflammation is not specific to these four symptom domains. If you can address neuroinflammation, what is believed based on the current literature, it can address by and large.
So could we consider that addressing neuroinflammation something as a characteristic of next generation?
I believe the distinguished characteristic. Yes, you can call it as a next generation should address this neuroinflammation, because one thing to remember, the schizophrenia psychotic condition or neuropsychiatric, it's not limited to only schizophrenia. If you look at movement disorders, Alzheimer's, major component of Alzheimer's disease is a psychosis. Currently available, there are no, to my knowledge, there is no new drug approved to treat to psychosis in Alzheimer's. Rather, off-labelly, currently available treatments are used for addressing this psychosis in Alzheimer's.
Okay, and those are probably intended for some Parkinson's maybe?
No, even in the Parkinson conditions as well, psychosis is one of the major conditions in this. Yeah. There is to my knowledge, there is only one drug got approved for the treatment of psychosis in Parkinson, that is Nuplazid, the Acadia, the inventor of that drug. So other than that, currently, there is no approved treatment for psychosis in Parkinson. So addressing the neuroinflammation and then improving the safety profile, especially the cardiac safety, is extremely important because these are vulnerable patient population, elderly patients. They take multiple medications.
And they're taking it over a long period of time, too.
Long.
So
It's a
the
lifelong
safety is a really,
treatment.
Yeah, safety is a really important thing.
Cardiac is, addressing cardiac side effects is extremely important in treatment, whether it is a schizophrenia or, most importantly, neurodegenerative diseases, Alzheimer's and Parkinson.
So there's another competitor out there working in developing late-stage clinical trials, Karuna, and they're working on something, and I believe they reported out some of their data. I think if you interpret it, if you look at it, the positive, they were successful addressing positive symptoms, but maybe less so on negative symptoms. What are the implications of that? And I think they're the only other one out there, I guess, that's really kind of competing with you, for approval in the near term. But what do you think of the data they reported, and how does that compare to what we've seen from Brilaroxazine so far?
Maybe I can share my thoughts on mechanism of action in general rather than the Karuna's data interpretation.
Sure.
So in general, if you look at, there is a kind of a general perception that dopamine serotonin target may not be novel enough to address the symptom domains. But if you look at the mechanism, biology behind the schizophrenia is primarily caused by imbalance of dopamine, serotonin neurotransmitters in the brain. The companies may have a different approach to balance those dopamine, serotonin in the brain, but to have an effective treatment, the goal should be balancing those two neurotransmitters in the brain. So now, in the past, in the last 20 years, major companies especially focused on developing a non-dopamine serotonin targeted drugs such as glutamate, like bitopertin from Roche is a very classic example, and then GABA receptor targeted compound, and then Merck and other companies extensively worked on that target. And then in the last decade, nicotinic receptors also is another target used. Now it's the muscarinic receptors. The whole baseline or the mechanism, biology behind this one is to targeting these downstream targets directly, allosterically or indirectly. It should balance the dopamine or serotonin or both in the brain to have the effect on schizophrenia. But in reality, in the past, with all these drugs, newer mechanism what I highlighted, whether it is a glutamate, GABA, or nicotinic, showed kind of a weaker efficacy in the clinical trial in the phase 2 or in the phase 3, when they try to increase the dose to have a clinically meaningful efficacy to allosterically modulate the dopamine serotonin level in the brain, that ended up in side effects caused by the primary target, whether it is a glutamate, GABA, or nicotinic. Now coming to the muscarinic receptors, so this is no different than, to my knowledge, than other glutamate, GABA, in principle, the approach for treatment. So muscarinic receptors, primary receptor has its own off-target activities for the side effects, GI side effect, cardiometabolic side effects. So by targeting this one, when one has to address those side effects as well.
Right. So does that mean they have to take another drug? Do they have to take like a combination?
Like if you look at a Karuna Therapeutics drug, it is a combination of two drugs, xanomeline and then trospium.
Okay.
So the trospium is used to mitigate the GI side effect.
Oh, okay. So that muscarinic activity is actually trying to be counteracted by the.
So the key to that one is, at a therapeutic dose, without causing cardiometabolic side effect, can we achieve the efficacy by balancing dopamine, serotonin in the brain? To my knowledge, the pharmacology-wise, there are clinical literature or scientific literature. Allosteric modulation lead to dopamine receptor modulation. By targeting muscarinic receptors, you can modulate the dopamine receptors or dopamine level in the brain. That can lead to PAN score reduction. But modulating the serotonin receptors to balance the serotonin level in the brain to achieve the negative symptoms or a mood symptom treatment or cognitive dysfunction, that could be quite challenging. So that's why any target what we use other than dopamine serotonin, it should deliver balancing the dopamine serotonin in the brain enough to have efficacy for major comorbid conditions. To my knowledge, so far, all this downstream target that what we believe addressed to some extent, some downstream targeted drug addressed balancing the serotonin level in the brain, not optimally dopamine. Some balanced dopamine level, not optimally serotonin level. If a drug can't balance both, treatment becomes incomplete. So there are other drugs to balance either dopamine or serotonin suboptimally, but the next generation should have, to a great extent, address dopamine serotonin level both in the brain while having minimal off-target activities. That's the challenge in developing a new drug. We believe the Brilaroxazine, based on the data, we were able to address both this to a great extent.
Yeah. One of the things that has emerged over the last decades is just the understanding of the genome, genetic sequencing and things like that, and identifying the sources of disease genetically.
Yes.
So what role do you think that plays in terms of developing new schizophrenia drugs, and also perhaps diagnosing patients or perhaps giving them the right drug and things like that? What role does understanding the genetic profile play there?
That's very important. The drugs currently in the market, including the recently approved Caplita, lumateperone, or even Corona Therapeutics, xanomeline. So these drugs, the IND, original development, happened prior to 2000. The genome sequence done completion, the knowledge of, to a great extent, genomes made available in the 2000. And then sometime in early 2000, around 2000 to 2010, there were many new receptors, human cloned receptors became available because they are extremely important to fine tune the drug properties early on. Our drug discovered in 2009. So
you had the benefit of that a little bit,
Correct.
I guess, understanding more receptors.
So we fine-tuned our drug molecule to have those effector, higher activity, antagonist activity for 5-HT2B and then 7 that has an anti-inflammatory effect. And then another receptors what we fine-tuned is a dopamine D4 activity. That is also extremely important to address chronic conditions, negative symptom, mood, and then cognitive. So that's the advantage with us while developing the drug. And then second thing is the CYP activities, human cloned CYPs, like
CYP3A4, right?
2D6 and other CYP species. That in human cloned CYPs were not available prior to 2000. So we fine-tuned the metabolic profile of our drug to overcome the CYP2D6. Unfortunately, there are many widely used antipsychotics, antidepressant drugs, and then for chronic conditions, primarily cleared by 2D6. This 2D6 is not a most abundant liver enzyme. And then drug primarily cleared by 2D6 can cause variety of drug-drug interaction side effect. So with us, we fine-tune the molecule not to go through the primary cleavage of a metabolism through CYP2D6. So that is another advantage with our drug that significantly contributed to drug safety.
So it sounds like the genetic understanding that we have has really helped refine the process of identifying new products.
So that's what I often tell. The drug, all dopamine serotonin targeted drugs are not same. So we need to see what are the improvement made in this drug by addressing these dopamine serotonin receptors. That information not available the drugs currently in a market when they were developed. So that's the key difference.
Yeah. Well, that's great. Build on the shoulders of others with the new knowledge and get a new product out there that really addresses the unmet need, right?
Yeah, yeah.
All right. Well, Dr. Bhat, thank you so much.
Thank you.
Always a pleasure to talk and get into the details of drug and drug development. I thank you for that.
Thank you.
All right.
Yeah.